I am Raghavendran, Bioinformatics Mentor and Research Consultant with OmicsLogic. Let me brief you a little bit about my background and research interests. I'm a computational biologist by training, and I specialize in the subjects like multi-omics data analysis and data science methods in precision oncology and protein structural bioinformatics. I completed my formal education in biophysics and bioinformatics and then graduated PhD in computational biology from Max Planck Institute, Germany. Then I worked on projects in the Institute of Bioinformatics and Applied Biotechnology, Bengaluru, and Jawaharlal Nehru University as a postdoctoral research scientist. Throughout my research career, I developed special interests and expanded my expertise in precision oncology through data science methods that is applied to several multi-omics data. So right throughout this career, right from my PhD period, I've had the fortunate experience to mentor different undergraduate students and enjoy the experience immensely. So I feel very much privileged to work with some of the bright young minds like you all in the past and also in the future. I wish and I hope you all to enjoy as much as I do in this partnership between Amity Noida and Pine Biotech USA. Now, let me briefly speak about one of my research projects on precision oncology. So I chose to, okay, it's doing, okay, great. I chose to speak specifically about this study because it utilizes a pipeline of very well established multi-omics data analysis methods on publicly available data. So I hope at the end of this small talk, this project will inspire you all to develop your own scientific curiosity, leveraging the abundance of data that are publicly available. So I think you all had a very good introduction about what cancer is and how that is the leading cause of deaths, disease deaths worldwide. So for such a dreadful disease, systematic cancer treatments have relied on chemotherapeutic drugs that are marginally more toxic to cancer cells than normal cells. This is a huge problem because on an average, only 25% of the cancer patients respond to cancer treatments. And in comparison, about 80% of the patients will respond positively to popular painkillers that are available in the market. So a prominent solution to this problem is precision oncological therapy, where a treatment strategy is designed by targeting the molecular characteristics of the patient's tumor. Hence, the treatment is personalized to the patients who are likely to respond positively to specific cancer therapies. Unfortunately, there is not enough data from cancer patients to molecularly characterize all the tumors under a multitude of different treatment options. Hence, we have to resort to a suitable model system to predict tumor responses to several treatments. So we have already covered why we have to do this, and let me straight away go to how. The tumor cells from cancer patients are subjected to next-gen sequencing techniques, and the information about the gene expression, gene mutation, and epigenetic variation were extracted. This data molecularly characterizes the tumors. These tumor samples are then grafted into immunosuppressed mouse and administered several cancer drugs. Then the tumor's response to these respective drugs are documented. This data tells us about the tumor's reaction to different treatments. Both the molecular characterization data and the treatment's response data are available along with the publication. Such a wealth of data generates a plethora of research questions. I use this data to design and implement machine learning models to predict tumor responses to different treatments that are available. So what did we find? We found that the treatment response could be predicted more accurately when we use data from multiple genes compared to traditional single gene biomarkers. And the accuracy of such prediction can be increased immensely with more than one type of sequencing data, such as the gene expression or the gene mutation or the copy number variation data was used to train the machine learning models. So that is what is depicted here. As we go from left to right, the information content increases. First, this method uses only a single gene biomarker, and the accuracy, which is supposed to be close to 1, is scattered all around the scale. But when we increase the information content to multiple genes and supplement that with the several other sequencing data like gene mutation data and copy number variation data, the accuracy tremendously increases and the consistency also increases. So what more could be done with this data? A gene set enrichment analysis on the set of most informative genes from these models can be performed to identify specific biomarkers for the tumor's response to different treatment regimes. One can also extend such an analysis to include patient data from the TCGA repository. And these efforts are currently being carried out by my partners in the JNU. So hope this was very informative and inspiring for you all to generate interesting research questions on the data that are publicly available. So I see that you had an excellent introduction to the platform that is OmicsLogic. And now I want to provide you a sample of how the lessons involving coding is organized on this platform. The courses that involve coding, a small assembly of courses that involve coding are given in this slide, and they're all designed and directed specifically towards students who have completed the introductory courses in their respective subjects. So all these courses have the following structure, and in the part that involve executing R or Python codes. So these lessons include what analysis are we doing, why are we doing such an analysis, and what do we expect to learn from such an analysis, and finally, how to do such an analysis. The codes themselves, as given in an example here, include sufficient explanatory comments at each step, so it is very easy to follow. And finally, when the output is generated from performing such an analysis, and we also give a brief account on how to interpret this analysis, how to interpret the results of such an analysis with respect to the biological question at hand. So this is how the coding lessons involving coding are handled in the platform that you are about to enjoy. So many students congregating at one place would naturally precipitate into an evolution of interesting student clubs. So Sciomics Club is one such initiative taken by all the students associated with Biotech. The goal of this club is to help fellow students to learn about bioinformatics research using or via group sessions and easy to take courses and debates and discussion. This club welcomes students from all the fields of life sciences and is not limited to anybody who has an introduction to bioinformatics or any such computational biology. So these students will meet together and learn from and with each other. So they have this club has definitive topics of interest every month, and based on this topic, 2 guest speakers are invited to talk on the topics of the month, which is then followed by short Q&A focused on the role of bioinformatics and some career prospects for today's students. And there are specific instructions on how to join and be part of this club in the OmicsLogic platform website. And I would like you all to know that I will be talking in detail as a guest speaker on this club this Saturday, that is November 20.